Quantitative Big Imaging, Lecture 8, Part 1, Uncertainty, Statistics and Reproducibility. Today's lecture will be split in 3 parts. They are pre-recorded because my business trip that I'm doing next week. So this is the only lecture we'll have for April the 18th. As usual, we load a bunch of modules, and today there are not really many special modules except for the ones I wrote and which are provided in the GitHub repository as you can download it. And there are also a lot of literature. As we are talking about statistics, there are very many books about statistics. There's also here a couple of books around uncertainties and how to handle uncertainties in image processing. Further on, we have also some papers and sites that you can visit. Some are Wikipedia, others are more about general ideas about, for example, the error propagation, how to handle that. And for fun, actually some of these videos, they are more serious, while others, like Last Week Tonight with John Oliver, is more for fun, but most have a twist that makes us think about what can be said with statistics if we handle it in the wrong way. The last one is a reflection on what can happen if you have uncertainties or numerical uncertainties in your programs. And apparently there is a safe case when you make a jump in Minecraft and sit in a boat, you will not die, except for on specific places. And that is what's reviewed in stand-up math. Otherwise, there are also further slides or websites that you can read on. Previously on the QBI lectures, we have gone through quite a lot already. So we looked at image enhancement, highlighting the contrast of interest in images, minimizing noise, understanding image histograms, looking at segmentation methods, automatic or semi-automatic, labeling different components and analyzing these components. And last week we also looked at more complicated shapes that we can look at. For example, if you have very thin structures or voids, then you can look at the skeleton and do analysis using that. Or you can also, if you have touching grains in or items, you can use the watershed segmentation. So by now we have both ways to extract structures from the images and also to do some analysis of these shapes, what they contain of. Today we are starting with some motivation as usually, having some scientific goals, going for reproducibility, and actually I see here now that this slide miss one part, and that's actually the part about uncertainty, which is really important. And that is what we are going to talk about the first part today. Then we look at statistical metrics and results, and further on, what is the effect of different parameterizations and how to look at the results in the end. So we have looked at a lot of things in the imaging course now already, but the thing we haven't really mentioned is the big factor. So what does big mean? So it could be not just even large, it means really that we are ready for big data. That means we have to be ready to handle volume, velocities and variety. And that should be handled scalable and fast, and should also even be easy to customize. So what do we do with the big imaging? We look at scientific studies to get a few or single numbers out of your data and make sure that these numbers are describing the structures well, which we already talked about. And now today we are going to look more into making these numbers scientifically meaningful. And the questions we are approaching is how to compare number from different samples and groups and compare them, see if there are differences within each group or not, and also between different samples. We can also compare different processing steps and see what is the effect on the analysis when we do so. And with that, we can find the best parameter selection for doing a robust analysis. And finally, we are going into also how to visualize all the data and how to handle the visualization. So let's start off what we have done. We are looking, for example, at our original cell image, and we got rid of the noise and found the cells, which were essentially lecture 2 till 4. We did analyze the shapes of the cells, which was lecture 5. And then we managed to separate cells that were touching or joined using the watershed from lecture 6. And we even looked at some metrics to characterize the distributions. And now we have at least some samples or regions in our images, and with that we get a large number of metrics and almost as large number of parameters to tune. So now is the question how we can do something with it. And the first thing is actually to decide what kind of experiment you want to do with all this knowledge. So there are different things that we want to look at. The two first ones we are looking at is the correlation between different parameters, and that means there is a statistical relationship between input and output or between different groups, and it's usually quite easy to show with a single calculation. Causation is a little bit more tricky. That means that there is a connection between A and B, and the question is what happens if we change parameter A and on parameter B. And it can be very difficult to show, and sometimes it's even impossible to prove that there is a causation. But experimentally, we can create experiments that we can show these effects. To do this, we have also different strategies on how we design our experiments. So we can have observational experiments and we can have control experiments. And the observational experiment means that we usually are exploring large data sets to look for trends in them. So someone else collected a lot of data and the population is essentially random. And this kind of analysis is not always hypothesis driven, and it's very hard to prove or show any causation. So some examples of observational experiments is that we, for example, examined 100 people, and the ones with blue eyes were on average 10 centimeters taller. Another example would be in 100 cake samples, we found a 0.9 correlation between baking time and bubble size. So these are examples that we just have a lot of data, and then from that we try to make conclusions. It's, on the other hand, more reliable or more relevant to use control experiments, because then you can say and already have an expectation of what you will get for a kind of outcome. So most scientific studies, they fall in this category. And by designing the experiment in a way that you can control what are the input groups, you can, in principle, also show the causation. So examples of the control experiments are like we have 50 mice with the gene XYZ switched off, and 50 mice with the gene XYZ on. And it showed that with this gene on, we had a foot size that increased by 10%. Another one is we increased the temperature, and the number of pores in the metal cast increased by 10%. So here we can see that there is a direct connection between the changed input parameter and some other observed parameter. Next is we want to look at also what kind of analysis we want to do. And initially we may do qualitative analysis, just observe and make conclusions from that. But that is usually just a pre-conclusion. And then we have the quantitative, where we really go in and measure different features or quantities within the data. And typically we are looking at using visual feedback, using rendering programs for 3D data or something. And we can compare our expectations with results from independent techniques and approaches. And if there are artifacts, is something we can see with the qualitative assessment. So just state the fact that, okay, we have a lot of artifacts, we may need to do something to clean them. We can also make the assessment if the shapes we have observed look correct, and if they don't, then we may go back to step one again and do a new experiment if we can. And also further on, if the different shapes are distributed as expected, and if they are oriented in the right way. So there are a lot of things we can say just by looking at the images and without putting any number on it. And the experience you have of the different material, you can also use there to make your assessment. But this is just a preliminary step on the way to doing the quantitative metrics. And here we can calculate different metrics, for example, the specific shape or distribution of a collection of metrics from the sample. And afterwards, it should be possible with each parameter to establish a relationship between the parameter and the metric, and in that way even produce a model that can predict what the process or the sample that you have observed. Now, doing all these measurements, we have unfortunately some uncertainties, and we even have uncertainties in image processing. And in principle, the question is, why do we have to talk about the uncertain? And as Richard Feynman stated in his book, Surely You're Joking, Mr. Feynman, scientific knowledge is a body of statements of varying degree of certainty, some most unsure, some nearly sure, but none is absolutely certain. And with that, we come to the conclusion that we can never say that we have an exact value, because there is always some kind of uncertainty in the instrumentation we have used. Could be that you use a caliper, which has some uncertainty. You have a voltmeter that measures the voltage within some uncertainty, and so on. So every instrumentation has an uncertainty, and that is what we want to build in to our analysis and show how much how precise we have measured. So what we want, in principle, it's easy to put error bars on things. If you just look at the statistical variation in the data, that is one thing. Then you can produce some kind of confidence interval, and often is based on the noise or sometimes in variations of the shapes. But we have more uncertainties than that. And as I just mentioned, distance measurements have their uncertainties. and other uncertainties are also included. And the problem is that uncertainty propagation in image processing is not really obvious. So we have to look for ways to handle it and get as close as possible. And on the topic of error bars, XKCD has a nice view of how to work with uncertainties. So it's often people talk about errors in your measurement, but it's actually usually what we want or can measure is the uncertainty. So the error is the absolute difference between a very precise known value and some deviating measurement. And for that we need a ground truth, otherwise we can't measure it. The uncertainty, on the other hand, is that you have an interval, you have your central estimated value plus minus the uncertainty. And this is more what we usually can handle. And the error is somewhere within the uncertainty region. Then which kind of uncertainties are we dealing with? We have technical uncertainties, so we have the choice of metric, which may be more or less precise. We have noise. There is unsharpness in the images. And on top of that, we can have effects that are introduced by the image processing. And a small contribution can even be numerical errors. It can be rounding errors, for example. And in addition to that, we have the field-specific uncertainties, which can be model simplifications or variations in the population that we are studying, or maybe forgotten parts of the population, which also introduce some uncertainty in our modeling. There are also different categories of uncertainties. So we have systematic uncertainties. They are reproducible and there doesn't need to be any statistical analysis in the same sense. So we have the rounding errors. We can have an uncalibrated system that provides a bias. Even the choice of algorithm can introduce a systematic error in our analysis. Then we have the random uncertainty, which is based on statistical fluctuations. So there can be natural variations in the studied population, fluctuations in the source driving the measurement system, and there can also be detector noise. So this measurement system can introduce some extra noise into the measurement. And all this builds up together into the final uncertainty that we are looking at. And typically the uncertainty can be described using these target practicing boards. So in this matrix we have the combination of accuracy and precision. And up in the top left corner we have low accuracy and low precision. So it's off from the center and it's also quite spread. And the opposite is in the lower right corner, where we have high precision, and in the high precision there we have instead a very well-centered and very densely packed targets. So these are the two cases, or two of the extreme cases, and in between we can also have low precision but high accuracy, so we are centering around the center target. And we can have high precision but low accuracy, then we're off, but we are very precise off. This can be translated into statistical terms. Then we're talking about bias and variance. So up in the left corner now we have large bias and large variance. And in the lower right corner we have small variance and small bias. And these are the terms that we can use in our description of our uncertainties. There are also the question how to measure the uncertainty depends on what kind of analysis we are doing. And here I have categorized it into counting and measuring. For the counting, we have discrete countable items, can be regions, can be pixels, or something like that. And it's always absolute integers that we have. And the uncertainties you have here, I mean, you are counting a finite number of elements. So the uncertainties actually come from preparations coming to the place where you can do this counting. So for example, we have the area of a segmented region, which is just counting the pixels that you have in that region. But the question is, how well was it segmented? How uncertain is that information? When we go to the measuring, we look at physical quantities, and then each value has some uncertainty. And these uncertainties, they are introduced by noise from the measurement system or variation in the sample, and it can also be caused by the instrumentation. And here we can, for example, look at mixing ratios extracted from the gray level of the image. The absolute and relative uncertainties is also something we need to look into, because we have to compare our uncertainties and errors from a neutral perspective. And let's say we can measure something with an uncertainty of 1 millimeter. And then we compare that to the distance to the moon, which is almost 400,000 kilometers, or the diameter of a coin. And that is quite a big difference, in particular if we can measure this with 1 millimeter uncertainty. And of course, if you look at the relative uncertainty, it's quite a small number for the distance to the moon, while it's actually 5% when you compare it to the diameter of a coin. So it's important to know what the relative uncertainty is, because not always that the absolute uncertainty tell you so much. And from the measurement theory, we have the propagation of uncertainty, and that equation is based on the derivative of the function and times the uncertainty. And that can be propagated if you do have a combination of variables. Then you have the effect of the sum of the two variables, and if they are uncorrelated, you can drop the last term here. But if you don't have it uncorrelated, then you have to measure also the cross correlation between the two variables. And in imaging experiments, we have some uncertainty that we can look at. One is the pixel size. We have how well the segmentation was done. Goes back to how you set your threshold or something else. Then you have sampling timestamps in time series of images. And of course, you finally also have the uncertainty in the gray levels. So some examples that I'm going to show you now. The first one is about determining the pixel size, because that's not always exactly known. And to measure the perimeter of some object. And finally, to measure water volume behind a pixel. Looking at the measurement of the pixel size, we need some things for our analysis. So first of all, we need an object with known length, and we need an image of the object made with the imaging system that you are going to use. And the pixel size is the object length in the metric object length divided by number of pixels between two edges of the object. And the uncertainty equation for that is that we have the uncertainty for A over B, and the relative uncertainty then turns into this equation. And if you want to look into it in more detail, there is this tutorial that you can look at. And yeah, you're welcome to take a look at it. So for our measurements, we have our object. For neutron imaging, we use gadolinium as an absorbing object. It has a very high attenuation coefficient. And this object, we know the distance, or more or less, we can measure it with a caliper. And the image that is caused by it looks like this. So you have a very black block here. And now we need to measure the distance between the edges, also with in pixels. This is in the tutorial that I mentioned. And our measurements with the caliper was 20 millimeters, and the uncertainty of the caliper is 0.05 millimeters. Then we have the pixel distance between those 2 points, and that is measured with to be 464.9 pixels, and the uncertainty from that was 0.32. And this we can now go on and put in our uncertainty equation. And you see here now that we have the uncertainty for the pixels and the uncertainty for the measured length. And this we put up here now in this Jupyter cell and compute it. So we have the different lengths here. Compute the pixel size and compute the relative uncertainty. And finally we print it, and then we can see that the pixel size in this case is 43 microns plus minus 0.25% approximately. And now we go on looking at measurements in segmented images. And that is something we did already in lecture 5, where we measured area, perimeter, positions, distances, etc. And at that time we considered them to be absolute values, and we just mainly count pixels. But how does the pixel size uncertainty mix into these equations or into these metrics? First, we have the perimeter length, which has 3 sources of uncertainty. First, how is the edge of the pixels identified? So that is now an algorithmic choice, how to extract the perimeter. Then we have the pixel size that we measured already, and its uncertainty even. And finally, how well was the image segmented? And the edges are actually the part that is segmented with the least confidence, as we will see soon. So, the algorithmic choice how to measure the edge pixels, there are different ways of doing it. The very simple way is that you just have the image, the segmented image, minus the erosion using some structure element. And that provides us with the pixels that belong to the edge. But the question is how now we handle these pixels. So the simple way is just counting all of them, and then we come up with 18. What we could do instead is we look at edges facing the background, and then we just counting these edges. And that brings us to a number 24, which is quite a lot more than the 18. Something in between would be to look at the center line. So then we look at the constellations of pixels on the perimeter. So this would be just a straight line. Then we have this the full length. And then we have here, we have half the distance plus half the square root of 2. And so on. And with that we come to a line length of 20.5, which is probably a better approximation than any of the other two. You could even go further and doing some kind of local fitting to the shape, but that wouldn't do much more precise than the 20.5. So this is the simple way of getting something that is more or less okay. And putting this into the uncertainty equation, so let's first assume we can trust the segmentation. The edge is 18 pixels long, and then we have a pixel size of 43 microns with some uncertainty. And we plug this into the uncertainty equation. And with that, we get that, that we, first of all, that we have the pixel size, or uncertainty in the pixel size, which is 0.11 microns. And then we have the fluctuation in the uncertainty in the edge length, which is this 0.11 times square root of 18. And then we come to the uncertainty of the length is half a micron. And the total edge length would then be 774 microns plus minus a half, which is not so big variation actually. The final example is to measure water volume from gray levels. And here we go into using the gray levels, or the variations in gray levels, and connect them to Beer-Lambert's law. And that requires that we know not only the pixel size, but also the attenuation coefficient for water. And from there on, we can extract this thickness. And with that, we get the volume behind each pixel. There are, of course, a lot of fluctuations because the image is noisy. But now we have the uncertainties, the standard deviation in the transmission, some confidence interval, and we, of course, have pixel size again, which has some uncertainty. This can now be put together. Again, let's trust we have done a good segmentation, and we have the thickness, which is D, and that is with some uncertainty. And then we have a pixel size, which is some size. And the volume equation for uncertainty is that we now have the pixel size squared times D. And this gives us the relative uncertainty of the pixel size squared times 4, and the thickness uncertainty. And that gives us then that the volume is here equal to 0.09 cubic millimeters and an uncertainty of 0.5% approximately. So these were some examples how you can propagate the errors in different images. And here I didn't even scratch on the whole processing chain of filtering and segmentation and anything of that. These are things I actually have not material for. It's rarely done actually. And there is a PhD thesis which handles it in the notes. And leaving that part, we can go for the segmentation. And this is inspired by Peter Moonen's talk at the ICTMS 2019. And there are different ways to evaluate the segmentation algorithm performance that we looked at in lectures 2, 4, and 5. So we looked at confusion matrices, we looked at ROC curves, we looked at heat maps. But all these, they require some kind of ground truth to say something. And what if we now want to look at what the performance is of the segmentation without having a ground truth? And the first thing is that we need to do is to assume Gaussian noise. So if we have an image with some noise, like here, then we can actually get the noise histogram, and from there we can estimate the shape of a Gaussian probability density function. And when we put the threshold between two Gaussian classes, then we also from lecture 4 learned that we have true positives and true negatives, And we also have, and that is the tricky part, we have also false negatives and false positives. And these are the ones that make our quality of segmentation worse. So what we need to do is, the first thing is we want to have a probability mix of classes for each grade level. The first thing we need to do is to find the probability density function for each class, and that can be done using an algorithm called Gaussian mixture model. And then we need to normalize the Gaussians to set either the peak to 1 or area on the curve to 1, but I actually prefer setting the peak to 1. And then for each grade level, compute weights. And the weights, they are computed using these Gaussians. The confidence for class I in this case is that we have the weight for the class I divided by the sum of all weights that are crossing or approaching this place. And with that, we can create a confidence map using the segmented image and select CI for each pixel. Now, what are the weights? Let's look at it. In the algorithm, we have this WI, which is the Gaussian distribution for observation at point X. And now we have an observation at this place here. And for class 0, which is the blue one, we get this value of W. And for class 1, we get this value. And now we can see for the confidence value of class 1, we get the confidence of about a third. And for class 1, we have the confidence of almost 90%. So it's clearly that we are more confident as we are closer to the peak of this distribution compared to the distance to the peak of that distribution. Looking at the three-phase segmentation, so we have in this case 3 classes with average intensity -3, 1, and 5. And then we produce noise with these different average values, and for all a sigma of 1.0. To make it a little bit more realistic, we also add some sharpness, so we get this smooth image where we have a smooth transition between the classes. And on top of that, we now add the noise. And the combination now, we want to segment. And looking at the segmentation, which I actually did some cheating, I just used the pattern for the classes that I used for generating the image. We could have done a lot of segmentation methods, but in this case, it's not about which segmentation method we are using. So let's look at confidence of class 0. Then we can see here that in these 2 regions, we are mostly confident that it is actually class 0. And that's because we actually are in it. And then there is actually some uncertainties in it because of the noise amplitude. Then in the region that should have been class 1, we have some uncertainty points that could have been, but it's not. And what you can see here also along the edges, which were smooth, we also less certain. Then we move on to confidence of class 1. Class 1 is the one that is here in the middle. And here you can see that you are actually in between, you're overlapping with 2 classes here. And that is what you see here, because in this place, this is the problem I referred to in the multi-class segmentation in one of the earlier lectures, that it could be that you have this transition region that is classified in the wrong way. And otherwise, yeah, there is some leaking into the other ones because of the noise amplitude. And finally, we have class 2. And again, we have the uncertainty around the edges. There is some leak into that area, but into class 0, there is essentially nothing. Because if you look at the confidence curve for class 2, there is more or less no overlap with the class 0. Combining all this together, we get an overall confidence map. And then we see that in this area is not so good, but in particular around the edges, in all cases, we have some more uncertainty in which value it should be. So this can be used in a way to tell how well we have segmented our images, and that can be then forwarded into our analysis of the data. So when does it matter that we have this kind of map, or to see what we're doing or how confident we are? And in a high signal to noise ratio image, then we have the lowest confidence near edges, and that has most impact on the perimeter measurements, on tortuosity measurements, connectivity, network content connectivity, and also for volumes of small items. If we now have a low signal to noise ratio image, then the low confidence is on all pixels actually, and then all the problems that we had for high signal to noise ratio, they apply to low as well. But we also have problems for measuring the porosity or volumes and for labeling. So it's, as usual, low signal to noise ratio is what we should avoid. And now we come to the end of the uncertainty part. So in principle, the conclusion is that the scientific analysis requires that you declare your uncertainties. And measuring uncertainties in imaging is not straightforward. We have physical uncertainties that can be quantified. We have noise that can be measured. Then we have the precision of how well the analysis can detect things in the image. And that also depends on the object to pixel size ratio. If you have very small objects compared to the pixel size, then you do a larger error than if you have large objects. And the effect of the processing can be quantified through simulations. So then you need to do some simulation experiments in order to extract some more knowledge about what are the effects of your analysis. This was the end of part 1 of lecture 8.